Well, it's wonderful to have this opportunity to talk about my research that's focused on optimizing the nutritional strategies to promote growth in newborns. And I'd like to thank ILSI North America for giving me the opportunity to be a scientific advisor and for talking today. So this is my disclosure statement. I'm editor-in-chief of the Journal of Nutrition, which is the oldest nutrition research journal in the world. I'm on the board of directors of the American Society of Animal Science, and my research is currently supported by the National Institutes of Health, the USDA, and one of the studies that I'm going to talk about today was supported by Abbott Nutrition. So there are many infants that are born in this country that are born of a low birth weight, which is defined as being born weighing less than 2,500 grams or 5.5 pounds. In fact, low birth weight infants constitute almost 10% of the infants that are born in this country and 15 to 20% worldwide. So that's more than 20 million infants that are born each year. Most of these infants are born preterm, which is being born less than 37 weeks of gestation. And this is a huge global problem. In fact, preterm birth is now the leading cause of child death. So this is a tremendous problem. And there have been an improvement in the survival of these low birth weight infants in recent years due to neonatal technologies and drug treatments, but morbidity still remains high. So obviously, optimizing the nutritional management of low birth weight infants is really crucial for not only their immediate health, but also their long-term health and well-being. Most of these infants are discharged from the hospital, weighing less than the 10th percentile for intrauterine growth standards. Most of these infants, or many of them, remain small to adulthood. And they exhibit long-term developmental outcomes, which we know are detrimental. So there's cognitive deficits that have been measured in some of these low birth weight infants. There's motor impairment, which can result in reduced work capacity, lower muscle strength, and mass. Low birth weight infants also can have a higher fat to lean body mass ratio. So there's an increased risk for obesity, insulin resistance, type 2 diabetes. There's also increased risk for metabolic syndrome and cardiovascular disease. So extrauterine growth restriction of low birth weight infants is frequently due to our inability to provide adequate nutrition to these infants. Now, because low birth weight infants are frequently feeding intolerant, we initially feed them by parenteral nutrition, which is providing nutrition by IV. And then enteral nutrition is gradually advanced over days to weeks until about 80% of enteral feeds are tolerated, and then the TPN is discontinued. So many of these infants that are fed enterally are fed by orogastric tube because they can't suck, swallow, or breathe normally. So when they're orogastric tube fed, they can be fed human milk or they can be fed formula, and they're fed either on an intermittent bolus schedule or they're fed continuously. So when they're bolus fed, then this elicits this pulsatile pattern of hormones and substrates in the blood. But when they're continuously fed, the insulin and hormones and substrate levels are very low and constant. So we wanted to know which feeding pattern was better because clinical studies have not been able to discern which feeding pattern is better in promoting growth, and that's, understandable because of confounding factors that are present in human studies. So because ethical considerations limit human infant studies, which Dr. Donovan has talked about, we have been employing the neonatal piglet model as a model for looking at the effects of nutrition on the regulation of growth. And indeed, the piglet model is a really good model for studying or modeling the human infant, because the anatomy and the metabolism of the neonatal pig is very similar to the human infant, and its body size is large enough, that it avails itself to a number of experimental approaches which you couldn't use in a mouse pup because they're so very tiny. So in a neonatal pig, you can repeatedly sample blood, you can do tracer kinetics across different tissue beds, and you can feed them different ways. You can feed them continuously, you can feed them bolus feeding, you can feed them parenterally or enterally, and of course, you can vary the composition of the diet. And that's really hard to do with studies that are done in mouse pups. So our studies in the neonatal pig have shown that ingestion of food causes an increase in the synthesis of muscle protein. And this increase in the synthesis of proteins in muscle is due to the rise in insulin in the blood and the rise in amino acids in the blood. And this insulin and amino acids then signal down to stimulate muscle protein synthesis. So just to give an overview of this, we know from studies that have been conducted in our lab that ingestion of food causes a rise in insulin in the blood. And this insulin then signals down the insulin signaling pathway, which stimulates the activation of mTOR, which is mechanistic target of rapamycin. So mTOR is this master protein kinase that it's a regulator of several processes in muscle, but it's a regulator of protein synthesis. So when mTOR becomes activated, it activates this translation initiation signaling pathway, which regulates the bonding of the messenger RNA to the ribosome, and together with the other translation initiation factors, elicits the synthesis of new protein. So we found in this newborn muscle that the abundance of many of these signaling proteins is greater than it is in animals that are just slightly older. But really quite interesting is that the rise in insulin in the blood that occurs after a meal elicits the activation of this signaling cascade. And the activation of many of these signaling components is much greater in newborn muscle than it is in muscle from slightly older animals. Now, we also know that when food is consumed, there's a rise in amino acids in the blood. And these amino acids signal down to mTOR, but actually how it does this continues to be a black box. So there are some purported signaling proteins that have been identified in recent years, and our lab has shown that the abundance of some of these signaling proteins is greater in newborn muscle. And the activation of these signaling proteins with the small rise in amino acids that occurs in the blood after a meal, the activation of some of these signaling proteins is greater in newborn muscle, like for the RAG proteins. So armed with this information, we asked the question whether bolus feeding might be more beneficial than continuous feeding, because we hypothesized that the bolus feeding would cause a more profound activation of the signaling components that regulate protein synthesis. So we took baby pigs and we fed them formula for 24 hours. And the formula was either given continuously at a constant flow rate, or it was given by bolus feeding, like a meal feed. And the meal feeds were given every 4 hours, and these animals received, the two different treatment groups, exactly the same amount of formula. That was a balanced formula to provide all of their nutrient requirements. And so what we found was that when the animals were continuously fed, that there's this low, constant level of insulin in the blood. But when they're bolus fed, there's this pulsatile rise in insulin just after the meal, and then that insulin falls down to baseline just before the next meal is given. And this is the same as the case for the amino acids. And so what this elicits is a greater increase in the activation of these signaling proteins that regulate translation initiation, and that produces a greater increase in the rate of protein synthesis within muscle. So this is what happens after a 24-hour period, but if we continue this for 21 days, what you can see quite clearly is that the body weight of these bolus-fed animals is greater than those that are continuously fed. And these animals were fed exactly the same amount of food, but yet the bolus-fed animals grew more. So they seem to be more efficient at converting their food for growth. Okay, so that's interesting that the body weight increased, but what's really important is what was the weight distributed toward fat, or was it distributed toward lean? And in fact, we found that lean gain was greater, and there really wasn't any different in fat mass. So this food is being partitioned more toward lean rather than fat. So I think this study clearly showed that bolus feeding causes this pulsatile rise in amino acids and insulin in the blood that activates these signaling pathways that stimulate protein synthesis and ultimately increases lean growth. But with continuous feeding, there is this lower level, constant level of amino acids and insulin in the blood that really rather mutes these signaling pathways and blunts protein synthesis and really restricts growth. So I think this study provided evidence that intermittent bolus feeding is the preferable feeding method for enhancing lean growth in infants that may be fed by gastric tube. However, there are some infants that are in hospital that have to be continuously fed because they're meal feeding intolerant. So what are we going to do to enhance the growth of these continuously fed infants? So because of other studies that we conducted in our lab, we turned our attention to leucine, because leucine is not only an essential amino acid, so it's a building block of protein like the other amino acids, but leucine is rather unique in that it acts as a signaling molecule that stimulates the pathways that regulate protein synthesis. And studies in our lab have shown that leucine seems to be the primary driver of that rapid increase in protein synthesis after a meal. So here's just an example of one of the studies that we've conducted that shows if you raise the leucine levels in the blood just up to the level that's seen with a regular meal, that you see an increase in the rate of protein synthesis. And if you give a similar dose of isoleucine and valine, you really don't have an effect. And this increase in protein synthesis is driven by this activation of translation initiation. So what we hypothesize is that if we gave a leucine supplement in a pulsatile manner during continuous feeding, that might enhance growth of these continuously fed infants by basically mimicking that pulse in leucine that normally occurs after a meal. So we took these baby pigs and we fed them a formula continuously by orogastric tube for 21 days. And these pigs were either pulsed with leucine or they were pulsed with alanine. So alanine is just the isonitrogenous control. And what we found was that when we pulsed with leucine, that we got an increase in body weight, even though these animals were fed exactly the same amount of food. And this increase in body weight was due to an increase in lean gain, and in fact, fat gain was reduced. So it seems like this pulsing with leucine caused a repartitioning of nutrients toward lean and away from fat. And this increase in lean gain was due to an increase in muscle weight that occurred in muscles of various fiber types from either the fast-twitch glycolytic longissimus dorsi muscle or the soleus muscle, which is more slow-twitch oxidative. And this increase in muscle weight was due to an increase in the rate of protein synthesis, which occurred in muscles of different fiber types and was due to an increase in the activation of the translation initiation factors that regulate protein synthesis. So I think this study provided evidence that pulsatile delivery of a leucine supplement during continuous feeding can activate the translation initiation factor pathway that increases protein synthesis and ultimately can enhance lean growth. Okay, so this is what happens with leucine supplementation, but are there other methods that we could use to enhance lean growth in the low birth weight or premature infant? So we turned our attention to some of the leucine metabolites. And one of the leucine metabolites that we looked at was beta-hydroxy beta-methylbutyrate, and this work was supported by Abbott Nutrition. So beta-hydroxy beta-methylbutyrate, or HMB, is normally produced in the body. So it's a lower concentration than what's in the blood that's compared to leucine. And it's beta-hydroxy beta-methylbutyrate is produced from an irreversible reaction with KIC. So it's a normal metabolite. So we chose HMB because there were some studies that suggest that HMB can promote lean gain and strength in adults, but there's also evidence to the contrary, and no studies had been conducted in neonates. So we took baby pigs and we either fasted overnight or we fed them a low-protein diet that was supplemented with different concentrations of HMB. Now, this lowest concentration of HMB is what's present in human breast milk. And this higher level of HMB was designed to reproduce the level in the blood that's seen when HMB has been supplemented to adult humans. And we fed them by gastric tube every 4 hours for 24 hours so we could exactly control the amount of food that they were consuming. And so here we found that HMB supplementation at the highest dose did increase the activation of translation initiation factors, actually up to the level that was seen with feeding a high-protein diet. And this resulted in an increase in protein synthesis that occurred in all muscles that we measured here, the longissimus dorsi, but it wasn't quite up to the level that was seen with feeding a high-protein diet. Now, immature muscle requires an increase in the number of myonuclei in order to support these highly anabolic processes that we've been talking about. So since myonuclei are postmitotic, they can't divide, we really need satellite cell proliferation in order to increase the myonuclei. So postnatal hypertrophy is dependent not only on increasing protein accretion through increasing the rate of protein synthesis, but we also need to increase myonuclear gain through satellite cell division. So it was very exciting in this study that even when given over a 24-hour period, that HMB supplementation increased satellite cell proliferation. So this has great, I think, importance in looking at the long-term effects of HMB supplementation in these neonates. And in fact, the increase in satellite cell proliferation was similar to what was seen with feeding a high-protein diet. So this is really exciting to show that HMB supplementation can not only increase protein synthesis, but also satellite cell proliferation. So, I think some of these studies that we've conducted over the past few years suggest that supplementation with leucine or its metabolite HMB might be useful to consider in the nutritional management of low birth weight infants to promote their growth. So on behalf of my lab, which involves a number of postdoctoral fellows over the last few years. I'd like to thank you for your attention.
Questions for Dr. Davis?
Thanks, Teresa. Lovely to see piglet research again. Takes me back. And it's really hard to do these studies, everybody should know, because those piglets are very vulnerable. What do you think the practical application is of, shooting up the babies with a little bit of leucine throughout the day? Have you had any discussions with the neonatologists and just, regulatory, giving a single amino acid shot?
Yes, I think, when I talk to the neonatologists in hospital, they become rather excited about this work because, when you think of, this was continuous feeding that was given by orogastric tube, so enterally, but a lot of these babies are fed TPN, so they're fed continuously. So, and this may be part of the reason why babies that are fed TPN, don't grow as well. So I think they're very excited, and so, one of the considerations for use of this would be in short gut, where the babies don't grow as well and they're fed continuously frequently. So this could be maybe the initial application of this work. Thank you.
What about during the period where the infants are being fed continuously, parenterally?
Mm-hmm.
Could that benefit, do you think, from a pulsatile approach?
Chris, I think that this showing that continuous feeding produces a lower level of protein synthesis and a lower level of growth is really, due to this constant level of insulin and amino acid in the blood. So it doesn't matter how that constant level is produced, whether it's parenterally or enterally, you would get the same effect. So I think it could be a useful way of improving the growth of TPN-fed infants. So I think it has great potential, but of course it has to be tested.
And the other thing is your hypothesis is that the pulsatile feeding is because of the amino acid effect, but could it be that the continuous feeding is, because as you mentioned, insulin is another important driver, is causing insulin resistance? Have you tried clamping the animals, doing euglycemic clamps on the animals that were pulsatile or continuously fed to see if they've developed some insulin resistance to the effects on protein synthesis?
Right. So I think that would be a great experiment to do. We haven't done it yet, but we do insulin clamps, and we've been doing those for about 15 years in our lab. So I think that would be a wonderful experiment to do. We're actually doing some of the work now in the preterm pig, and that's one of the experiments that we're funded for from National Institutes of Health. But we haven't done the experiment yet.